Hello students, welcome back to my channel. Today I am going to start a new lecture series about genetics, and today my topic is about on search for the genetic material. Which one is the genetic material, DNA or RNA or protein? Today we have modern technologies, today we have many scientific knowledges about that DNA is genetic material, but long ago there was a debate among scientists level which one carry the genetic information, DNA or RNA or protein. We begin by recounting how scientists discovered the nature and structure of the genetic material. Long before DNA and RNA were known as genetic material, scientists realized that living organism contains some genetic substances, a genetic material that is responsible for the characteristics that are passed on from parent to child. Geneticists knew that the material responsible for hereditary information must have three characteristics. And which are that three characteristics? First of all, it must contain in a stable form the information about an organism, its structure, function, development, reproduction, anything. Second point, it must replicate accurately so that the progeny cells have the same genetic information as the parent cell. And point number 3, it must be capable of change. Without change, what happens? Without changes, the organism would be incapable of variation, and adaptation and evolution could not occur. So these are the three characters. The first successful experiment was done by Frederick Griffith in 1928. Frederick Griffith in 1928. Prior to that Griffith experiment, many scientific experiments were done to probe which one is genetic material. But the first remarkable experiment which was accepted by the scientists worldwide, and this experiment was Griffith's transformation experiment in 1928. Frederick Griffith, he was a British medical officer who worked with Streptococcus pneumonia. Streptococcus pneumonia bacteria. The Streptococcus pneumonia, as the name already indicates, it is responsible for causing pneumonia. Griffith used two strains of this bacterium, the S strain and R strain. Two strains of this bacteria. One is S strain and another is R strain. And which is the significance between calling them S strain and R strain? S for smooth and R for rough. This one is produced. First of all, we should have knowledge what is colony formation. In microbial techniques, then if we plated it on an agar plate and introduced the bacteria, they will produce colony. It is a general knowledge about microbiology. An agar plate is prepared and bacteria is added here and they will produce colony. And this A strain, this produce shiny colony, okay, and this R strain, this one produced rough structure colony. So what's the difference between them? This one produces smooth colony. This one produces rough colony. This one is virulent, means it has the power to cause pneumonia disease, and this one is non-virulent. Non-virulent, and A strain have this virulence is due to existence or due to presence of a polysaccharide coat is present, and here polysaccharide coat is absent. I have used PC for polysaccharide coat, short form polysaccharide coat. It is capsule. Polysaccharide coat is nothing but capsule surrounding each cell of bacterium. So this is difference between these two strain of Streptococcus pneumoniae, and this coat is also reason for appearing as shiny smooth appearance of S colony, smooth colony strain. R strain and S strain, they are not completely different, but here R strain have a mutation which prevents them to form this polysaccharide coat, and hence this is non-virulent in nature. In this case, a mutation in gene affects the ability of the bacterium to make a coat, and hence alters the virulent state of the bacteria. Several types of A strain with different compositions of polysaccharide coat. Griffith worked with 2S and 3S strain. Griffith works with 2S and 3S strain. Type 2 and type 3. Type 2 and type 3 coats. Now come to Griffith experiment. What had Griffith done? Griffith injected mice with different strains and observed its effect on them. When mice were injected with 2R bacteria, 2R bacteria injection, mice, as it is not virulent, so mice is alive. And what is this 2R bacteria? It is mutation from 2S bacteria. When mice are injected with type 3S bacteria, injection, mice are dead. Obviously this is very normal. This one is non-virulent strain and this one is virulent strain, so mice live and dead. And after that what he performed is heat killed 3S. 3S heat killed. Injected into mice, and mice alive, because after the heat kill treatment the 3S bacteria all have dead now, and when they are injected into mice, the mice was alive. No pneumonia is observed, no pathogenic condition is observed when this 3S bacteria is heat killed now. But when they have injected with 3S heat-killed bacteria with 2R. All of we know that this one is non-virulent strain of bacteria. When injected with mice, the surprising incident that mice was dead. This one is heat killed, so it has no power to generate disease in mice. But 2R strain, it is also non-virulent. But when these two are mixed, the surprising observation was observed that mice is dead. So, what is the cause? So, must be there is something, or as all of we know, we have knowledge that when we hit the DNA, so there must be something which one is transferred, which one is going to modify this 2R strain into virulent strain. So what was that material? As all of we know that after giving heat, DNA, as all of we have that knowledge, the DNA become denatured. Denatured means only the H bonds between them were broken, and this one is called denaturation of DNA, and after the heat is withdrawn, the DNA renatured. Okay, but DNA structure is not break down so easily by heat, by applying heat only, but protein it is completely denatured. Protein structure is completely destructed when we apply heat. So this is heat killed bacteria. So must they have something alive in them. This is cell of R and S. This one is R2 and this one is bacterial chromosome, as both of these S3 bacterial chromosome. This one is bacterial chromosome. After the heat kill treatment, what is the fate of now S3? The naked DNA come out, and this bacterial cell wall is broken down by heat killed. Naked DNA is coming out. Okay, and what is the significance of this transformation experiment? Transformation is nothing but taking up. Transformation, transduction and conjugation. These are three methods of horizontal gene transfer processes found in bacteria. What is transformation? Transformation is nothing but taking up the naked DNA of another bacteria by another bacteria. So here the naked DNA is found after heat kill treatment of S3, and this naked DNA is uptaken by this R2 bacteria. Okay, what will be the fate of this bacteria now? If we can use this with a different ink, this is the bacteria different ink, and this one is now transmitted into. And become part of this. Okay, this is called the transformation process. So what is the transformation process? And this transformation process happening here, the DNA, the naked DNA of S3, heat killed S3 bacteria, the naked DNA is uptaken by R2 bacteria and become part of this, and this R2, which was non-virulent type, after uptaking this naked DNA from S3, now it become virulent and it causes the mice dead. Okay, so it must be DNA. So it must be DNA which can transfer the virulence of this S3 is now transmitted to this R2 bacteria. And after this experiment and this explanation, all of the scientific community will not going to accept the level, but till now it needs further research, and the research and experiments were going on. And after this 1928 30s, for a long time, 1930s to 1940s, Avery, MacLeod and McCarthy experiment were done, which will be discussed in my next class. So keep in touch.